You sit down outside. The sun is warm on your face and forearms. The brightness makes you squint, but the skin registers nothing beyond the warmth. No pain, no signal, no indication that anything has changed underneath the surface. You will stay for an hour. By the time you stand up, 7 physics pathways will have activated simultaneously inside your skin, each running on a different timeline, each invisible at the surface, each producing consequences that range from trillionths of a second to decades. Everything that matters during this hour happens while you feel nothing but warmth. Melanin absorbs photon energy in picoseconds. DNA damage forms in minutes. Vitamin D synthesis runs in parallel through the identical wavelength. Collagen crosslinks accumulate in the deeper dermis. Repair enzymes race the expanding count of broken code. The tanning response that would reduce the damage will not arrive for days, and the redness that tells you it was time to go inside will not appear for another 4 to 6 hours, long after every consequence that mattered has already been set in motion. The clock starts the moment ultraviolet photons reach the skin. What follows is the hour traced through every layer, from the melanin that catches the first photon to the repair enzymes that will still be working long after you go inside. Minutes 1 through 5. The fastest photoprotective chemistry in biology activates. Ultraviolet photons in the 290 to 400 nanometer range, invisible to your eyes, pass through the outer dead cell layer of the epidermis and reach the living cells below. Inside the melanocytes, the pigment-producing cells scattered through the basal layer, each photon strikes a melanin polymer. Melanin's molecular structure is a conjugated system with electrons delocalized across an extended network of carbon bonds, a molecular architecture specifically evolved to handle photon energy. When a UV photon hits this network, it excites an electron to a higher energy state. In most biological molecules, that excited state persists long enough to drive a chemical reaction, breaking a bond, creating a reactive radical, initiating a cascade of damage. In melanin, ultrafast internal conversion channels the electronic energy into molecular vibration, heat, in picoseconds, trillionths of a second, faster than any other energy conversion in human biology, faster than the chemical reactions the photon would have driven if it had reached any other molecule in the cell. The photon energy arrives as light and leaves as warmth before any chemistry has time to begin. Brenner and Heering characterized this process in photochemistry and photobiology, confirming that melanin converts UV photon energy to harmless thermal energy faster than any other photoprotective molecule in the human body. A photon that could have reached DNA and broken a thymine bond instead becomes a pulse of heat that dissipates into the surrounding tissue. The melanin absorbs, converts, and dissipates. The DNA behind it never knows the photon arrived. Each melanocyte distributes melanin granules to approximately 36 surrounding keratinocytes through branching extensions called dendrites. The granules, packaged inside structures called melanosomes, are transferred to the keratinocytes and positioned as physical caps over the nuclei, dark umbrellas shielding the DNA from photons arriving from above. One melanocyte protects 36 neighboring cells through this architecture. The architecture is deliberate. The melanosomes concentrate on the sun-facing side of the nucleus, the side where UV photons arrive, leaving the opposite side uncovered because no photons approach from below. The geometry of the shield matches the geometry of the threat. The melanin already in your skin when you sat down is the shield operating during this hour. The reinforcements the body is about to order will take days to arrive. And while the melanin shield intercepts photons at the surface, a second reaction runs in parallel through the same wavelength. Ultraviolet B photons, the shorter wavelength between 290 and 315 nanometers, reaching the epidermis strite molecules of 7-dehydrocholesterol sitting in keratinocyte cell membranes. The photon energy breaks a specific bond in the steroid ring structure, converting the molecule to pre-vitamin D3. Body heat converts the pre-vitamin to active vitamin D3 over the following hours. Hollick established the photobiology of this conversion in the New England Journal of Medicine, confirming that the molecule the skeleton and immune system depend on for calcium absorption and immune regulation is produced exclusively through the wavelength that also damages DNA. Here is the paradox built into the physics. The wavelength building your vitamin D is the identical wavelength that will be rewriting your DNA 3 minutes from now. Melanin cannot selectively block one while transmitting the other, because both reactions require photons at the same energy level. More melanin means more UV interception, more protection, and less vitamin D production. Darker skin produces less vitamin D per hour of sun exposure because the melanin shield that protects the DNA also intercepts the photon the vitamin pathway needs. One photon, two opposite biological consequences determined entirely by which molecule it strikes first. The paradox has no resolution in physics. The body cannot increase melanin to protect DNA without simultaneously reducing the vitamin D production that the skeleton and immune system require. Populations that evolved in high UV equatorial environments carry more melanin, more UV protection, and require longer sun exposure to produce adequate vitamin D when they move to higher latitudes. Populations that evolved in low UV northern environments carry less melanin, less protection, and produce vitamin D more efficiently from shorter exposures. The trade-off between protection and production is written into the melanin level of every human skin type, calibrated across thousands of generations to the UV intensity of the latitude where the population spent its evolutionary history. Melanin catches most photons. What reaches the DNA when it misses? 5 minutes in. Photons that pass the melanin umbrella reach the DNA. Photons at the shorter wavelength arriving at keratinocyte nuclei are absorbed directly by adjacent thymine bases on the DNA strand. The photon energy drives a cycloaddition reaction, forcing a covalent bond between two neighboring thymines and creating a cyclobutane pyrimidine dimer. The two bases that were reading independently are now locked together by a chemical bridge that should not exist. The DNA strand kinks at the dimer site. The genetic code at that position can no longer be read correctly by the enzymes that transcribe it. Repair activates within minutes. Nucleotide excision repair, the pathway the CT scan discussion described, racing to fix X-ray damage, detects the distortion the dimer produces in the helix. A recognition complex identifies the kink by scanning the helix for structural abnormalities, sliding along the backbone until it encounters the bend the dimer created. Once found, a helicase enzyme unwinds the DNA around the damaged site, exposing the dimer for removal. Two endonucleases cut the damaged strand on both sides of the dimer, excising approximately 24 to 32 nucleotides containing the fused thymines. DNA polymerase fills the gap using the intact opposite strand as a template, reading the undamaged code and rebuilding the excised section base by base. Ligase seals the backbone, the dimer is gone, the code restored, the strand intact. One repair event from detection to completion takes minutes to hours depending on the location in the genome and the accessibility of the damaged site. Some regions of the genome are tightly packed, the DNA wound around histone proteins in dense coils that the repair machinery must unwind before reaching the dimer. Other regions are open and accessible, the repair complex arriving and operating without obstruction. The repair time varies with the terrain. Sankar mapped the full kinetics of this pathway in his Nobel Prize research, establishing that a single dimer can be detected within minutes and excised within hours under normal conditions. During these early minutes, the repair enzymes handle the incoming damage efficiently. Every dimer that forms is met by an enzyme that removes it. The queue of unrepaired dimers stays short because the processing speed exceeds the arrival rate. You feel nothing at the surface. No redness, no pain, no signal that DNA is being simultaneously broken and repaired underneath the warmth. The repair system evolved across billions of years of solar exposure. For the first 15 minutes it keeps pace. 15 minutes carry more weight now that I know where the repair race begins. The physics running beneath the surface is entirely invisible, entirely silent, entirely absent from the warmth the skin reports. But the clock started the moment the first dimer formed, and every subsequent minute adds to the uncleared load. What arrives in 2 days is already being ordered. And if the reinforcements take longer to arrive than the attack takes to land, minutes, 15 through 30. The keratinocytes damaged by UV release a signaling molecule called alpha-melanocyte stimulating hormone. This signaling molecule binds to receptors on the melanocytes, activating the enzyme cascade that synthesizes new melanin. The tanning response has been ordered. Delivery spans days. The enzyme tyrosinase must hydroxylate the amino acid tyrosine to a melanin precursor, then oxidize the precursor to dopaquinone. The dopaquinone polymerizes into melanin through a series of intermediate steps, each requiring time and enzymatic processing. The completed melanin polymer must be packaged into melanosomes. The melanosomes must be transferred through dendritic extensions to surrounding keratinocytes. The keratinocytes must position them as nuclear caps. 48 to 72 hours from signal to functional deployment. 2 to 3 full days between the order and the delivery. Thursday's tan does not exist during Tuesday's exposure. Every minute of this hour is defended by the melanin that was present before you sat down, the baseline level determined by genetics and prior exposure history. You cannot accelerate the timeline. The enzyme cascade runs at its own speed regardless of the urgency of the UV signal that triggered it. The gap between the attack and the reinforcement is 2 to 3 days wide, and every hour of sun exposure in that gap is spent behind a shield that was built for last week's conditions. You are spending this entire hour behind yesterday's melanin, and tomorrow's melanin will not arrive until the day after tomorrow. This is why the first strong sun exposure after months indoors produces the worst damage. The melanin baseline is at its lowest. The repair enzymes are at resting capacity. The tan that would reduce next week's dimer load is still being manufactured from today's UV signal. Every layer of the defense system is running on reserves that were calibrated for indoor light levels. Now, the longer ultraviolet wavelengths penetrate deeper, 30 minutes in. Ultraviolet A photons, the longer wavelength between 315 and 400 nanometers, carry less energy per photon than the shorter wavelength but penetrate past the epidermis into the dermis, the deeper layer where the structural scaffolding of the skin lives. Inside dermal fibroblasts, the longer wavelength generates reactive oxygen species, superoxide radicals, hydroxyl radicals, singlet oxygen. These are molecules the body produces during normal metabolism, but it generates them at a rate and concentration the antioxidant defenses in the dermis cannot fully neutralize. Radicals from the longer wavelength attack collagen fibers, the long triple helix proteins that give skin its tensile strength and its ability to snap back after stretching. Oxidative crosslinking welds adjacent collagen fibers together at points where they should remain separate. Stiffening the matrix the way glue between the pages of a book prevents the pages from moving independently. Elastic fibers, the proteins responsible for the recoil, fragment under the radical assault. The extracellular matrix that provides skin with its resilience stiffens and degrades simultaneously, losing both strength and elasticity through the same hour of longer wavelength exposure. You can see the cumulative result of this chemistry on your own body. Hold the back of your hand next to the inside of your upper arm. Both surfaces carry collagen laid down at one age, one genetic blueprint, one developmental program. The texture, the elasticity, the depth and pattern of the wrinkling are dramatically different. The inside of the upper arm has been shielded from UV for your entire life, by clothing and by the arm's natural position against the body. The back of the hand has received decades of direct, longer wavelength exposure during every outdoor hour. Solar elastosis, the leathery, deeply lined texture of chronically sun-exposed skin, is the collagen cross-linking chemistry made visible on a surface you can compare against its own protected control, every day, on your own body. The experiment has already been run. The results are on your hands. The longer wavelength that drove the crosslinking arrived at wavelengths the SPF number on the sunscreen bottle does not measure, penetrating deeper than the shorter wavelength that formed the dimers, operating through a different chemistry on a different tissue layer, producing damage that accumulates without any repair pathway capable of reversing it. Collagen crosslinks are permanent. Each hour adds to a total that only increases. For 30 minutes, the defense has been keeping pace with the damage. The melanin intercepts most photons, the repair enzymes process most dimers, and the system is working. But the balance between damage and repair is not static. Each additional minute adds dimers to a backlog that the enzymes process at a fixed speed. The dimer arrival rate is proportional to UV intensity, which has not changed. The enzyme processing rate is proportional to the number of repair complexes available, which is fixed by the cell's protein production capacity. The ratio between arrival and processing tilts with every passing minute, and the tilt only goes in one direction. What happens when the damage rate exceeds the repair rate? Minutes 45 through 60. The repair race tips. Cyclobutane, pyrimidine dimers are now accumulating faster than the nucleotide excision repair enzymes can process them. The repair workload has exceeded the enzyme capacity. For the first 30 minutes, every dimer that formed was matched by an enzyme that removed it. The arrival rate and the processing rate were in balance. By minute 45, the arrival rate has pulled ahead. Unrepaired dimers persist in the genome through DNA replication. When the polymerase reaches a dimer it cannot read, it stalls at the fused thymines, unable to determine what the original sequence was, and must proceed by inserting a base without template guidance. The inserted base may match the original, it may not. Each unrepaired dimer is a probabilistic event, a coin toss between accurate bypass and permanent mutation. Most mutations land in the 98% of the genome that codes for nothing critical. Silent passengers copied forward without consequence through every subsequent cell division. Some land in the 2% that includes tumor suppressor genes and growth regulators, the genetic territory where a single base change can disable a checkpoint that prevents uncontrolled growth. While a protein called p53 surveys the cumulative damage load across the genome of each cell. Every cell in the epidermis is running this assessment independently, each one tallying the dimers its repair enzymes have processed and the dimers still waiting for repair. Two verdicts are possible. If the damage is repairable, 53 halts cell division. The cell cycle arrests, granting additional time for the repair enzymes to work through the damage still waiting without the pressure of the approaching replication deadline. The cell survives with its genome restored. The arrest may last hours, the enzymes clearing the final dimers while the cell sits idle, waiting for the all-clear signal that allows division to resume. If the damage exceeds the repair capacity, p53 orders apoptosis. Caspase enzymes activate in a cascade that dismantles the cell from the inside. The DNA is fragmented deliberately, cut into pieces too small to replicate. The cell membrane blebs, forming small bubbles that neighboring cells and immune cells recognize as debris for recycling. The cell dies in a controlled sequence designed to prevent a corrupted genome from ever dividing again. Brash demonstrated this UV-triggered apoptosis pathway in the Journal of Investigative Dermatology, establishing p53 as the molecular judge that determines whether a UV-damaged cell lives to divide or dies to protect the organism. Sunburn peel arriving in 3 to 5 days is mass 53-directed apoptosis, millions of keratinocytes executing the self-destruct command issued during this hour. The sheets of peeling skin are dead cells being shed after p53 determined their genomes were beyond saving. The peel is the body disposing of cells it concluded were too dangerous to keep, the visible evidence of a molecular triage that ran beneath the surface while you felt nothing but warmth. I checked the time differently after learning this sequence. 45 minutes in the sun is where awareness should sharpen, where the repair system's capacity to keep pace with the arriving damage reaches its limit. The hour that felt like rest was the hour the balance shifted. 60 minutes pass and you go inside feeling fine. The damage finished an hour ago. Why does the redness arrive only tonight? The skin is warm, perhaps slightly pink, but nothing that registers as damage. The physics continues without you. 4 to 6 hours after UV exposure, the redness arrives. Prostaglandins synthesized from damaged cell membranes by cyclooxygenase enzymes. Cytokines released from injured keratinocytes signaling distress to the immune system. Histamine from dermal mast cells, the same molecule responsible for the swelling and itch of an allergic reaction, now deployed in response to UV tissue damage. Blood vessels in the dermis dilate, increasing blood flow to the damaged area and producing the visible redness that you interpret as sunburn. Plasma leaks from the dilated capillaries into surrounding tissue, creating the edema, the swelling, the taut tenderness that makes the skin painful to touch. Redness is the inflammatory response to damage that occurred hours earlier. By the time the sunburn appears, every dimer that would form has already formed. Every melanin shield has already activated. Every repair enzyme is deep in the unrepaired count. The inflammatory cascade that produces the visible redness requires hours to synthesize the prostaglandins, release the cytokines, and dilate the vasculature. If you returned from the beach saying you did not burn, you were already damaged at the beach. The inflammatory alarm took the full delay to mobilize. The redness is the body's delayed report on an event that ended before the report was written. The damage and the alarm system operate on different clocks, and the alarm clock runs hours behind. This delay is why the most dangerous UV exposure is the one that feels comfortable. The skin sends no real-time warning that damage is accumulating. The only signal during the hour is warmth, and warmth is pleasant. The redness that would have told you to go inside arrives when you are already home, already showered, already hours past the point where leaving the sun would have changed the outcome. The alarm system operates on a delay that is longer than most exposures. You sat in the sun for an hour and felt nothing concerning. 6 hours later, the mirror shows the consequence. The damage was complete before the alarm was assembled. The report arrived after the event was over, and by the time you see the redness, the repair enzymes have been working through their backlog for hours. The P53 assessments are already underway, and the cells whose genomes were too damaged to save have already received their apoptosis orders. The sunburn you see tonight is the body's retrospective announcement of physics that finished this afternoon. Well, how long does the damage from 1 hour last? This hour deposited dimers on cells that have been absorbing UV photons for every hour of sun exposure across an entire lifetime. The unrepaired dimers from this afternoon join the unrepaired dimers from last summer, from a decade ago, from the childhood sunburn at the beach 30 years past. One unrepaired dimer is one hit on a multi-hit model that requires several independent mutations before a cell transforms. The second hit arrives from another hour, another year, another photon on a different afternoon that you will never connect to the first. The third may come from a replication error during normal cell division, the polymerase misreading a base at a site weakened by a dimer that was repaired imperfectly years earlier. The cancer that appears at 65 is the integrated product of every hour in the sun across a lifetime, untraceable to any single afternoon, accumulated across thousands of individual exposures that each contributed 1 or 2 unrepaired dimers to a running total that no medical record tracks and no diagnostic test can decompose into its individual contributions. Mutations accumulate the way mileage accumulates on an engine. No single kilometer causes the failure. The failure is the sum of every kilometer driven, and the cells that accumulated the highest mileage are the ones that did the most work. Cells that absorbed the most photons across those decades are the melanocytes, the protector cells that shielded every neighboring keratinocyte by intercepting UV into their own bodies. Each melanocyte sits at the center of a unit of 36 keratinocytes, absorbing photons that would otherwise reach the nuclei of the cells it protects. The melanocyte absorbs so that the keratinocyte does not. This is its function. This is also its vulnerability. Moles are dense clusters of these protectors, more melanocytes per square millimeter than surrounding skin, more UV absorbed per hour, more protection delivered to surrounding tissue, more photons intercepted into the protector's own genome. A mole that has been on your skin since childhood has been absorbing UV on behalf of its neighborhood for decades. Shane and Bastian traced the genetic progression in Nature Reviews Cancer. After decades of UV service, one melanocyte accumulates enough driver mutations to transform. An initiating mutation in a growth signaling gene, present in approximately 80% of common moles, activates a growth signal. The mutation alone is not sufficient for transformation because 53 is still functional, still surveying damage, still ordering cell cycle arrest or apoptosis when the genome is compromised. Then P53 loss disables the damage checkpoint. The molecular judge that spent decades ordering damaged cells to stop dividing is itself silenced by a UV-induced mutation. Then a third mutation, loss of the third gene, removes the growth brake entirely. 3 independent mutations, accumulated across years from separate exposures, converging in one cell. After 30 years, the cell that protected your skin becomes the most aggressive transformation in oncology. The protector absorbed the most because it protected the most. My attention to changing moles sharpened the day I understood this paradox. The cell that shielded everything around it carried the highest cumulative load of the photons it intercepted across every hour of sun exposure, across an entire life. The mole is a monument to decades of service, and the service came at a molecular cost that only accumulates. One final point of physics remains. Sun protection factor, the SPF number on the bottle, follows an asymptotic curve that the number on the bottle conceals. SPF 15 blocks 93.3% of the shorter burning wavelength. SPF 30 blocks 96.7%. SPF 50 blocks 98%. SPF 100 blocks 99%. The jump from 30 to 100 adds 2.3 percentage points of shorter wavelength protection. The number on the bottle tripled. The protection increased by a fraction of what the first application of SPF 30 already provided. Meaningful protection is from 0 to 30, removing nearly 97% of incident shorter wavelength photons and reducing the dimer formation rate proportionally. Every SPF unit after 30 captures a smaller and smaller fraction of the remaining 3%, approaching but never reaching 100% blockage. The curve flattens toward an asymptote that no SPF number can cross because some shorter wavelength photons will always penetrate the sunscreen film regardless of concentration. If you reach for the highest number on the shelf, you are paying for a marginal improvement the physics cannot deliver. The meaningful decision was already made when you chose to apply sunscreen at all. And the SPF number measures the shorter burning wavelength only. The longer wavelengths that drove the collagen crosslinking at minute 30, the longer wavelength photons that penetrate into the dermis and generate the reactive oxygen species responsible for the photoaging visible on the back of your hand, are not captured in that number. A bottle labeled SPF 100 may block 99% of the shorter wavelength that causes dimers while allowing the longer wavelength that causes collagen crosslinks to pass through largely unimpeded unless the formulation specifically includes longer wavelength absorbers listed separately on the label. The number that feels protective measures only half of the relevant physics. The physics started in picoseconds and the consequences run for decades. The hour ended and you went inside. The repair enzymes are still working through the backlog. The inflammatory response has not yet begun its mobilization. The tan is 2 days from deployment. The collagen crosslinks will never reverse. The melanocyte carrying the heaviest load may transform across a timeline longer than you will track. What did the body tell you while all of this was happening? Warmth. That was the only signal it sent to the surface.